We're a toxicology lab, and we focus primarily on what we call toxicogenomics, and that means that we look at how animals respond at the gene expression level. And in the last decade, we've developed these tools called RNA-seq that allow us to look at all of the genes in an organism simultaneously. And that's obviously a really powerful technique, and a lot of people have used it in a lot of different applications. And my lab used a lot of this as both the NRDA, the National Resource Damage Assessment process that we were a part of, and also as a part of our earlier GoMRI grant. And my co-PI on this grant, Marisol Sepulveda from Purdue University, was also involved in that. And so between the two of our labs, we had over 150 of these RNA-seq libraries, we call them. So it's a really large, really powerful data set where fish were exposed to oil under different conditions. And the premise of this project is that we can do meta-analyses of these libraries to identify not just responses that are specific to one organism, one life stage, or one oil exposure, but are in common to larger sets, perhaps all different fish in general, or different particular types of organism. And so we're comparing these transcriptional libraries to identify which particular pathways that the genes are involved in might be affected communally by oil. So we've actually found some really cool things, and we had a couple of talks here at this session and a poster. And the two things that we're focusing primarily on are that the idea that cholesterol biosynthesis is one of the most commonly affected pathways. We've seen that a lot of the other groups that are doing this sort of thing have reported it as well. But the fish seem to respond differently. Some fish have this pathway activated, some fish have this pathway deactivated. This is a pathway that's tremendously important in a lot of downstream processes. So understanding why these pathways are affected in the way that they are is really important. And so we were able to use these libraries to identify what we think is the proximate mechanism that's causing this up or down activation of these pathways, which then lets us predict downstream what the organismal effects are going to be. The second one is that we can look at the immune system of the fish and identify that the particular components of the immune system are being affected communally across different fish species. So in some fish species, we see all the pathways are being activated in exactly the same way, and that lets us be able to predict with some degree of certainty how they're going to respond to an oil with and without a pathogen stressor. So there's a couple of answers to that. There's kind of the basic science answer, which is what I just gave you, which is that we're trying to understand at a very basic cellular molecular level how these organisms are responding when they're exposed to contaminants under various conditions. And that helps us understand again, at a very basic level, what's going on with these fish. And if we know that, then we can predict out to other circumstances to understand how the fish might respond. At a more applied level, it lets us understand what's happening when we take the fish at the individual level, the cell level, and then we can extrapolate that out to the organism and population levels. And that's obviously a much bigger leap, and we're focused primarily on the basic science side. But if we can make that leap, then it provides us with a lot of power to say, look, if you have fish that are exposed to an oil spill in an area where they're exposed to hypoxia or pathogens or things like that simultaneously, these might be the effects that you would see that you wouldn't expect if you looked only for the predicted effects under oil exposure alone. What we always want is we want the ability to a priori predict what the effects of an event are going to be. And so we've got a long history in ecotoxicology of trying to understand what molecular markers we can use that'll tell us what the effects are going to be in other exposures and later on in life. And so that's basically what we're trying to do, is try to understand what the basic processes are that allow us to understand what's happening at the whole organism or population level.